Today we will be discussing the key findings of Nirenberg and Matthaei's experiment in 1961. We will also look into Nirenberg and Leder's experiment in 1964. Both of these experiments were crucial for the discovery of the genetic code. Here we have a chart depicting how Nirenberg and Matthaei found the codons that corresponded to the amino acids for each nucleotide base. They focused on RNA, which does not include thymine, so this was excluded from the experiment and replaced with uracil. They found that UUU encodes for phenylalanine, CCC codes for proline, and AAA encodes for lysine. However, after multiple attempts, they were unsuccessful in coding GGG. Nirenberg and Matthaei identified the amino acid specified by an RNA homopolymer. A homopolymer is a short strand of mRNA with one nucleotide. Adenine, guanine, cytosine, and uracil underwent polynucleotide phosphorylase to create homopolymer of each base. These poly-mRNA were added to a test tube with cell-free extract and incubated at 37 degrees Celsius so that translation could occur. This procedure had to be repeated 20 times to ensure all amino acids would be studied. The test tubes were then checked for radioactivity, which indicated which amino acid was associated with the homopolymer. In this case, the uracil homopolymer had a radioactive effect on phenylalanine. This meant that UUU encodes for the amino acid phenylalanine. This experiment was repeated with homopolymers of adenine, cytosine, and guanine. As previously stated, they were unable to encode poly-G homopolymer, but found CCC to encode for proline and AAA to encode lysine. Next, Nirenberg paired up with Leder in order to identify the 61 codons we know today. They used a technique that synthesized mRNA, tRNA, and a ribosome together. This was known as triplet binding. To the left, we have a structure of a tRNA. We will go in depth about the chart to the right. As shown, a trinucleotide, tRNA, with amino acids and a ribosome are mixed together to bind into one structure. The unbound tRNAs are then filtered out by a nucleocellulose filter, which is depicted here as a Brita filter. Nirenberg and Leder were able to find the 61 codons using this method by looking at the ribosome-bound mRNAs on the filter. For example, here the sequence GUU binded with valine and stayed on the filter. In addition, they found the 3 stop codons. In the end, it was thanks to these two experiments conducted by Nirenberg, Matthaei, and Leder that we have established the genetic code charts.